So I'm very happy to say again that it's a real pleasure to be here. So I thank John, Annie, Pat for the opportunity, and I'm delighted to be in this room because the future is in this room. Each of you have got an enormous responsibility in the future in terms of global security, in terms of food production. Each and every one of you here have decided to come to a very exciting, very stimulating course, obviously a very bright group, very committed people, and so I'm delighted to be here with you because the future in many ways is in your hands. It's pretty grim when we look into the future, and so each and every one of you have got an enormous responsibility to fulfill and to have a fantastic career and to contribute to global security, to contribute to in so many ways that we can. So it's a pleasure to be here with you. What I've got up here is probably a little bit like a field that Scott's got in the as a wheat breeder. It's a wheat breeder's field. It's like a microarray in many ways, isn't it? It's many thousands of different genotypes. Every single one of these genotypes is integrating what's going on in terms of temperature, rainfall, radiation, what's going on in the soil, and it's integrating it to produce a final yield. There are many different sorts of plots here, very large ones. Here is a plot harvester. So that little dot is a big plot harvester, harvesting those plots. So it shows the scale of a conventional breeding program. It's very, very powerful. Okay, why are we here and why is the future in this room? This is a very, very depressing figure. It was a New Scientist a couple of years ago, and it shows the average annual increase in yield in the last 10 years, which are these low values, compared to the 30 preceding years. So for our most important crops, for maize, for wheat and for rice, we're going backwards. We're not going forwards, we're going backwards. Yet our population is increasing dramatically. Our demand for carbohydrate through increased animal protein and so on is increasing at a very alarming rate. Our water consumption is being tapped off in all sorts of different directions, not just for agriculture, it's going into all sorts of other areas. But look at the yield progress in the last 10 years compared to what it was before. It's very, very depressing. Let's have a look at dryland agriculture. This is two countries where there is no irrigation available for water use. We have United Kingdom and we have Australia. And have a look at the rate of yield increase in the last 10 years. In the United Kingdom, where yield levels are up around the 8 tonnes per hectare, it's plateaued off. In Australia, which is very low yields purely because it's water limited. When was the highest yield? In the last the highest yield, tonnes per hectare over Australia was about 1996, 14 or 15 years ago. So our yields are plateauing exactly the same as what I showed before on that earlier graph. So each and every one of you in this room have a responsibility to really start to look intensively at how you can increase crop production, how you can secure food production into the future as we go forward, because at the moment it looks like we're stagnating. So we have to bring the best minds, the best ideas, the best science forward to really make a difference. breeding in dry environments is incredibly difficult. Have a look at what we contend with in Australia. This is the rainfall variability in New South Wales and Queensland, and it shows the yield every year. It zigzags up and down all over the place. You select in a very dry year, and the next year is a very wet year, and the yield progress is probably going to be zero. So the heritability for yield in an environment which is so variable, which all of our rainfed environments are, they're highly variable, the heritability for yield is close to zero. In other words, it's very, very difficult to make yield progress in these dry, fluctuating environments because of the large genotype by environment interactions. Now, this looks pretty exciting. This is the yield of durum wheat varieties in Western Canada compared to their relative to the year of release, and this is a percentage of Hercules. And you would think that the breeders who have been responsible, who were appointed, the breeders who were appointed back in 1970 should be winning a Nobel Prize. But it's the scale which is very, very misleading. They have been incredibly successful, there's no question. They've been very, very successful in making yield progress there, but when you calculate it out, it's only about 1% per year. And that is just not good enough when we go forward. We have to do better than this spectacular, or what looks to be a spectacular gain. We have to do better than this. Okay, we've talked about drought resistance, drought tolerance, water use efficiency. I far prefer to use water productivity. The problem with drought tolerance, drought resistance, is that there are no units. Well, what's the unit for drought tolerance? Is it percent survival? I've got no idea what the units are. If you are going to have a metric to beat, to get to improve on, it's much better to have a metric that you can actually measure and calculate. So I would prefer to use the word water productivity because you can measure it. The units, it can be rainfall or it could be, so on the bottom axis here, it's either rainfall or irrigation or water input. And then you've got grain yield. Doesn't have to be grain yield. If you're working in a perennial grass species, it might be total biomass. If you're working in sugarcane, it might be total sugar yield, or sugar beet, it might be something sugar yield again. So, but this is a metric where you can actually plot rainfall or water versus grain yield, and you get units which could be, in my terms, kilograms per hectare per millimetre. And every year you can see, last year if my yields were 15 kilograms per hectare, 15 kilograms per hectare per millimetre, in 5 years' time if I'm getting 20 kilograms per hectare per millimetre, it's a measurable value that you can estimate, you can see whether you're actually making progress. And this is the sort of values that farmers are more likely to be using, and we have to be talking to farmers to get our messages across, and we have to be talking the same language. So rather than using drought resistance and drought tolerance, which I have no idea what they really mean, let's use a measurement or a term like water productivity. It combines what we're aiming for in most instances. That's productivity, it's yield, it's biomass, whatever it might be, and it's related to how much water input there actually is. This figure shows pretty much where we are in Australia, and I don't think it's anything different to anywhere else in the world. So there is a current potential along this line here. For wheat, it's about 20 kilograms per hectare per millimetre. So if you know, if you had a season where there's only 150 millimetres of rainfall, you can only expect to get a yield of about 0.7 of a tonne. If you have a yield of about, if you have about 300 millimetres of rainfall and you top it up with a bit of irrigation water, then you might expect to get about 5 tonnes per hectare. So it's a very, very effective metric. But look where the current farmers are and look where our current potential is. There is quite a gap. How do we fill in that gap? The gap can be filled by better agronomy, a better physiological understanding of what the limiting factors are, and better genetics. When Monsanto executive makes a statement like this, the most important gene we have for drought resistance is, I sit up and take notice. So what did he say? The most important gene we have for drought resistance is resistance to root worm. The most important gene for drought resistance is resistance to rootworm. What's that all about? What's rootworm got to do with drought resistance? Why is that important? The first requirement when you're improving yield or improving productivity in water-limited conditions is to have a healthy root system. If you have rootworms that are nibbling away at your root systems, your root systems are going to be totally ineffective, and you can have the most incredible functioning above ground plant, but if it's got a lousy root system, it will be absolutely decimated. A good example of that is some work we've been doing with cereal cyst nematode. It is a very, very difficult trait to measure. This is a susceptible line, and this is a very resistant line. You could take this to a child in primary school to tell you which is going to be the most productive line from these, which is going to have the best performance. It's very difficult to phenotype. There is a fantastic molecular marker available, but you can just imagine how decimated plants or crops are that are susceptible, and how good the plants are which are resistant. So a healthy root system which is resistant or tolerant to the prevailing microorganisms in the soil or prevailing pathogens in the soil are going to have probably a bigger impact, as the Monsanto executive mentioned, than any other trait that we might be talking about, or any other trait that we've actually talked about over the last couple of days. So first of all, get a healthy root system, because that makes a massive difference in terms of performance in dry conditions. So I think this is a very, very important message in relation to improving performance in dry conditions. I think this is probably the first time this has been mentioned over the last couple of days, but you can see why it's so important. Another good example is certainly in Australia and in many other parts of the world, we have a really big problem with acid soils. And most plants are very, very susceptible to acid soils. Again, you can just imagine so these are near isogenic lines, are about 99% identical except for one gene. They're about backcross 8. So about 99% identical. This is their growth in an acid soil with and without a gene for acid soil tolerance. This is the performance or growth of the root systems in a non-acid soil. And again, you can see that the most important thing in terms of improving productivity in dry conditions is to have this gene for acid soil tolerance. This is in wheat. Fortunately, it's controlled by a single gene. It's a beautiful story. It's single dominant locus. It's been identified and cloned. It's a fascinating story. This is a root tip growing down through a soil. What happens is the aluminium, which is in the soil, which has actually sort of become much more available to the plant, or which is very, very toxic to the plant, becomes released when the soil is quite a low pH. And what it does is that the aluminium sort of prevents growth, but what this tolerant line does, the root system of this tolerant line, it exudes malate from the root system. And what malate does is sequester aluminium, and therefore it enables the root system to grow into these acid soils, to collect the nutrients, to collect the water, to use all the water in these very dry conditions. So again, root health is extremely important. Being able to tolerate chemical issues in the soil are very, very important if you're going to increase productivity in dry conditions. So that gap, which I showed before, and I think I probably show it again, can be filled by some of these solutions which I'm showing. So it sequesters aluminium. This final slide shows a transgenic barley plant. Barley is extremely susceptible to acid soils. You take the gene out of wheat, you put it into, you transform a typical barley plant, put the gene to wheat and grow them in an acid soil, and have a look at that incredible root system, which is going to be fully functional in terms of taking up water, taking up nutrients. So is this drought tolerance? We don't think of it as drought tolerance, but in terms of water productivity, it's very, very important. Okay, we've covered let's move now to real drought type issues. More drought resistance or more crop per drop? In Australia and probably certainly in the United States, most farmers probably wouldn't even harvest a crop like this. Why should we be working? Why should we be bothering to breed plants which are going to survive these dreadful, terrible droughts that we do get every now and again? Because even if you double the yield in these conditions, nothing is nothing. So the focus of our work perhaps should not be on these extremely dry conditions, but it's in conditions where the farmers are going to be able to make a profit, where they're going to be able to harvest the crop and continue its growth. So let's forget about some of these horrible, awful conditions, because there's nothing we can do about it. Plants must have water to grow. There is nothing that we can do about these plants here. We can make them survive a little bit longer, but they're not going to produce very much at all. Let's focus on the more productive areas. So water productivity. There are a couple of examples where we may be able to, in terms of root health, in terms of chemical toxicities in the soil, which may move us from here up to here. We probably have a potential or a goal to move even higher. I really believe that we can change the intercept here. I really believe that we can change the slope of this line so that we can go towards a completely new potential. And this is where we have to move. So how do we move to that new potential? There is no question that we're never, ever going to dispense with smart conventional breeding. This will always be the cornerstone of crop improvement. It'll always be the cornerstone of crop improvement. Do we concentrate on this and really get that right in terms of getting the numbers right, getting the efficiencies right, getting the rights of environments, getting the right genetics in there? Do we concentrate on this, or do we start taking a physiological approach? And most of the last couple of days have been spent thinking about the physiological approach. My career has been spent thinking about a physiological approach again. They're both important. They're both very important. Why, given the success of conventional breeding in the past, given the spectacular gains that have come from conventional breeding in the past, why should we embark on a physiological approach? Why should we be even having a go at that? Is it just intellectually satisfying? Is that the reason why we're doing it? Just to have good fun? Or is there something really achievable? I think there's something really achievable, and the reasons why I think this is important are for these reasons. First of all, taking the physiological approach, you can start to identify some of the limiting factors. You may not be able to identify, or you may not have the genetic variability in a breeding program for these limiting factors. So by taking a physiological approach, you can increase the genetic variability for the important traits. And if you don't have the genetic variability in a breeding program, you'll never make gains. You must have that genetic variability. Maybe you can make faster gains by selecting for a physiological trait than you can for yield itself. Yield has a heritability in Australia in some of these fluctuating environments of 0, so the genetic gain is very low. Whereas some of these physiological traits have got a very, very high heritability. Yield evaluation in a breeding program is not cheap. It costs me about $20 a plot. When I go out there and grow my thousands of plots in the field, it costs me about $20 a plot. So it's not cheap. And it takes many generations before you actually get enough seed to be able to grow a plot. So a yield evaluation is not cheap. And normally you have to do it, you have to replicate it, you normally have to replicate it across multiple environments as well. So maybe it could be more cost effective as well than yield evaluation, although you'll never get away from yield evaluation. Maybe you can do out of season selection. Maybe you can do 3 or 4 generations of selection a year. For yield, you can only do it once per year. You've only got one chance per year. For a physiological trait, if you really push through the generations, maybe you can get 2 or maybe 3. It's more amenable to marker-assisted selection, and you can also pyramid some of these traits as well. So there are quite a number of real, genuine advantages in terms of taking a physiological approach. But you have to know which traits are going to be the most important. This has been my mantra. John Passura is a wonderful colleague of mine who wrote about this in a very obscure journal quite a number of years ago, and it simplifies things enormously. It really simplifies things. Yield, this is always true. Grain yield, or total biomass production, is always a function of how much water the crop will get, how efficiently it uses this water and converts it into dry matter, and if it's a grain crop, how efficiently it converts it into harvest index. So if you are going to increase yield, one of these traits, one of these components must increase. So if your favourite trait or your favourite gene or your favourite transcription factor, if you cannot make a connection between your gene, your trait, your whatever it is, to either water use efficiency or harvest index, then my recommendation is to move on to a trait or a gene that does. Because that is the only way that you can make progress. Okay. For wheat, which is the area which I've been working on for quite a long time, we've identified quite a lot of traits which are going to be important. Some traits are going to be important in some environments, they're going to be worthless in others. Some traits are going to be much more universal. So traits are going to vary depending on the region, on the environment, on the crop, and this is a sort of selection of traits which which we've identified as being likely to have a genuine, real impact on crop improvement. I'll talk a little bit about some of these, but here we've got good establishment, increased vigor, carbon isotope discrimination, increased seed set during reproductive stage drought, presence of wax or pubescence, root systems, and so on. There's quite a range of traits which you can relate back to that pasture identity, and you can really see that there are compelling reasons why they're going to be important. So I'll give a couple of examples of those later on. But what are the elements of success when you've actually pinned down your most important trait? The elements of success are, first of all, effective phenotyping. And I'm going to come back to this on several times. Effective phenotyping is so important, not just for improving yield, but it's also just so important in terms of gene identification, QTL discovery, and so on as well. Effective phenotyping is absolutely of great importance. Effective phenotyping of large numbers of lines is so important. And I think I've probably spent more time thinking about how do I develop a really effective method of screening for a complex trait than almost anything else I've done. So it takes a lot of effort, a lot of thinking. The second element of success, apart from effective phenotyping, is getting out into the field and getting your boots dirty and evaluating your favourite traits, your favourite genes, whatever they are, in the target environment. So even with complex traits like root systems, we do a lot of coring of root systems to identify how deep the roots are. This is a bit of lab data. We're validating it in the field here. Coleoptile length here, but getting into the field to really validate it, because this is the only way to get proof of concept. This is the only way to convince the next layer of people who you probably relate to that whatever you've been doing is going to be very important. Field data is like, good field data is like gold. People listen to you when you've got that. And these are some of the conclusions I believe are the elements of success. So to be successful, you've got to be passionate. You have to have a real focus on what you're doing and not jumping around all over the place. You have to have a focus. Passionate focus, you have to collaborate. We've heard quite a bit today about teams required, but collaborate. You cannot, no single person can do everything. You have to be able to be using the skills of the person next to you, or in the next building, or the next city, or the next country, whatever it might be. Collaborate. It's so important. It's so much more effective. It's so much more fun to be talking to people about something that you're passionate about. Have a reality check every now and again by getting into the field or doing the critical experiments in the glasshouse or getting into the field. Have a reality check to make sure that you're on track. If you have a focus and if you are passionate and you have collaborations, then serendipity just seems to happen. It just seems to fall into place when all of these things, when you've got these things together. And then don't just let it sit there, but deliver. Write up the paper, get the information out, talk to the breeder or to the wherever you are in the hierarchy. Talk to the cell biologist or the plant physiologist or the plant breeder or the farmer or the commercializer or whatever it might be, but deliver, deliver, deliver. So these are what I feel are very, very important factors in terms of the elements of success. So again, let's get back to some of my favourite traits. They're specific to wheat. They probably have applicability to other crops as well, but my focus is my target environment is Australia. My target crop is wheat. That doesn't mean to say that these aren't important in other crops, in other countries either. I'll just go through a couple of examples of these, and I'll start off with carbon isotope discrimination. We've heard a bit about it today. It's very curious that when you look at the, and maybe it's just been a bit of a, it's a difficult thing to comprehend, it's a hard thing to understand, but basically when you look at the carbon composition in the air, which has got mainly carbon-12 and a little bit of carbon-13, and you look at that composition in the plants itself, they're quite different. So the plants actively discriminate against carbon-13. And this is the basis for the theory behind the relationship between carbon isotope discrimination and water use efficiency. There is a very good relationship between, so what we've done here to generate the data, and this is very important as well, you have to get to get compelling data about a trait. It's very difficult. You have to get basically near isogenic lines to do it. You have to get contrasts where the background genotype is basically the same, but the lines just differ for the trait or the gene itself. So it takes a while to actually develop this type of genetic material. When you have that genetic material, though, it's gold, because it'll tell you the value of the trait. So what we did with carbon isotope discrimination is that we established how to select for it. We select for it during the vegetative stage under well-watered conditions. It's very, very highly heritable. Even though it's controlled by many, many genes, it's very highly heritable. And when you take these near isogenic contrasts in the field in a particular genetic background, we found that there was about a 10% yield advantage in the driest environment, and it was pretty much neutral when you get up to the absolute high, very, very high yield levels. We were able to establish that it is a complex trait. It involves stomatal conductance, it involves photosynthetic capacity, so both of those are highly complex. There are about 9 genomic regions that come from the donor parent that's responsible for it, so quite complex. And so taking a QTL approach probably would not be very effective because there's 9 QTLs and they only account for about 50 to 60% of the total variation there. So the phenotypic approach in fact turns out to be more effective there in terms of breeding. And we've been able to incorporate it into some commercial varieties. Don't underestimate how important it is, or don't underestimate how important it is to move it into a commercial variety, but also don't underestimate how difficult it is to incorporate it into a commercial variety as well. The commercial world is vicious, it's hard, it's tough, it's demanding. They don't want a line with a high yield, they want a line. They want a wheat, say, that they can trade internationally. They want a wheat that Australia wants, a wheat that we can outcompete Canada for, or outcompete the United States for, or Turkey or some other country. So it's very, very competitive internationally. The farmers don't care whether the wheat is being traded at a competitive advantage or not, but the farmers are interested in a wheat which combines not only the trait, but the resistance to the diseases that they have in their field on a regular basis. So they want resistances to rust, they want resistance to nematodes, they want adaptation and so on. So it's very, very challenging to actually deliver on our traits. It's very, very difficult to get something which is competitive in the marketplace because the market is tough. But that's what we have to do. I'll take another example. Firstly, the enhanced early establishment, which is to do with new dwarfing genes, coleoptile length, I won't go into it now, and also enhanced vigor, which is important also. So 2 extra traits now, which I'll sort of I think it's the last example, which I think are very important in dry environments. If you don't get good establishment, then you don't get a good crop. you don't intercept much radiation, you get poor growth, you get low yield, you get poor delivery to farmers. For more vigorous early growth, there are many environments in Australia, and particularly Mediterranean ones, where about half of the rainfall that our farms receive, the rainfall just evaporates straight back into the atmosphere again. So half of the rainfall is used by the crop, and every millimetre of transpiration is growth and photosynthesis and biomass. So only half is used by the crop, and half of it is just lost from the soil surface straight back in the atmosphere. What a waste that is. Half the water not being used by the crop. If we could produce much more vigorous lines which shade the soil surface and reduce that soil evaporation, maybe you're going to be able to get 60% of the rainfall or 70% of the rainfall through the crop, through transpiration, through growth, through photosynthesis, increasing biomass, increasing yield, and only lose, say, 30 or 40% from the soil. We're never going to totally eliminate it, but we can make a big difference in terms of water productivity. So we've been very successful in being able to identify a whole set of new genes that are responsible for long coleoptiles, yet maintain the highly desirable plant stature in terms of height. We've been very, very successful in being able to double the leaf area of our plants, and in fact this has all been a very intellectually stimulating area, because the basis for this, we discovered that the reason why you can double the leaf area from here to here is through a change in the size of the embryo. Fancy the size of the embryo being responsible for a doubling of the leaf area. So something totally unexpected we came out with in this work. And this extra vigor is also associated with a difference in the leaf architecture of the plant. You can just imagine if you have a very, very thick leaf and suddenly you change the architecture of it and make it very, very thin, you've doubled the leaf area. So it's been very intellectually exciting to actually discover what some of these underlying factors are. Now, not only does an increase in leaf area reduce soil evaporation, but it reduces the size of the root system. We didn't know which way this was going to go, but we were delighted to find out that as we increase total above ground biomass, we're having this massive effect below ground as well. So we increase the size of the root system as we increase the above ground growth. Not only that, but by increasing the early leaf area and the early growth, we reduced the weed competition that was growing as well. So we reduced the weeds. As we increased leaf area, the above ground parts, we reduced the weed pressure as well. So again, increasing more water through the plant or through the crop than through weeds. And we increased nitrogen uptake. So this single factor of moving from a more vigorous plant has had a massive impact in a whole different sort of had a cascade of effects all the way through. Okay. These are the traits that we've been putting a lot of effort into. I've listed them on the left-hand panel here. And I want to look at them and consider what sort of selection environments do we use? Do we select for them under dry conditions, or do we select for them under favourable conditions? Do we have molecular markers for these? Or do we select for them using molecular markers, or do we select for them using the phenotype? So I've listed the traits down here, and let's look at the selection environment, the markers whether we've identified markers or genomic regions, and what is the most efficient method. When I assembled this, it surprised me a little bit more. I was aware of it clearly, but when I assembled it, it really did surprise me. For all of these traits, which I suggest are very, very important for wheat or cereals or timber crops in very dry environments. What is the most effective selection environment? It's not under dry conditions, it's under favourable conditions. Why is that? Partly it's because you can control favourable conditions much more easily. You're maximising the heritability. That's one important reason. The other important reason is that most of your growth occurs under favourable conditions. As soon as it gets dry, there's not much more growth that you have. So in all of the traits that we're focusing on, which we think are very important, the selection environment is under favourable conditions. We have identified molecular markers, or perfect markers in some cases, and we have identified genomic regions for every single one of these traits. So we're in a position to either use molecular markers for selection or to use the phenotype. What do we use? What is the most efficient selection method? In almost every instance, it's the phenotype. Now, that's what it is today. That doesn't mean to say it's going to be the same tomorrow or next month. Tomorrow or next month, as we improve our selection efficiencies, or as genomic methods get more effective, or when we get close to the gene, it could well be that the molecular selection could be more important. But at the moment, we are capturing far more of the total genetic variation with a relatively high heritability and fairly high throughput using the phenotype. So the summary of this table, firstly, accurate phenotyping is most important. Second important point is the traits are complex, and in many cases molecular markers or QTLs may not be very effective because they are complex. The easy traits have already been fixed. It's the complex traits that we're left with. To make gains under drought, we're probably going to be, it's more effective at the moment to select under favourable conditions, and validation in the field is critical, difficult and chancy. I was really surprised to read this statement by Craig Ventner in Nature back a couple of months ago. You think of Craig Venter as being this extraordinary person who's been able to mobilise this amazing effort to sequence the human genome, and just recently the development of this synthesis of a replicating cell, which is quite a remarkable thing. But his power in terms of the human genome has been quite extraordinary. But when I read this, that basically the effort that he's made in sequencing the human genome has got no value until there is phenotypic information, and it is no different to plants. So the phenotypes at the moment are representing a much greater challenge than are the genotypes. And crops are no different to humans in this respect. I won't go through this, but I was just going to quickly show ways of introgressing a trait, a complex trait, into a new variety. There are a number of ways of number of ways of doing it, but if you want more information about this, I'm happy to communicate through email with you about it. But it's very important if we are going to deliver, then we have to have breeding schemes to deliver by. And this is one that I think is one of the genuine opportunities to really deliver to breeding companies. But I will close with these key messages. So the future is in this room. I genuinely believe that. I think there's the enormous opportunities that are ahead. You are the people who are going to be responsible for making these gains in the future. We can double water productivity. The first couple of slides I showed were pretty grim and bleak, but we can double productivity. There is a big gap there, but we can make a big impact on that. It's going to require a combination of better genetics and better agronomy. And it's the combination of those two that's going to fill that gap and has got the potential to double productivity. But there are going to be no miracles. Progress is going to be very, very slow. There will be no miracles. They've all gone. Its progress will be slow. I think it's very important that we benchmark water use efficiency in the way I did with water productivity, so we can actually have a very good metric and focus on productivity and not drought resistance or drought tolerance. Smart conventional breeding will continue to be the cornerstone of improvement, and link up with your local breeder. Talk to your local breeder as much as you can to get the feedback, because it's that feedback of knowledge which is so important. The breeder is on the ground all the time. He's got his ears tuned to the industry. He knows what his plants are like. He knows what's going to be important in the future. So link up with your local breeder. The big challenge is still going to be phenotypic information and not genotypic information. And the future, I think we really have to develop some really bold initiatives. Where does the future in terms of water productivity go? In my work, I'm scaling down a lot of the above ground work that we've been doing, because I think we've reached a limit. I don't think we can go much further. We've got more to deliver yet. We've got to deliver to farmers, we've got to deliver new varieties, but I'm scaling down a lot of our above ground work. Where I think the future is what's going on below ground. I think we know so little about what's going on below ground, yet the morphology that we see above the ground is also reflected in the morphology which we cannot see below ground. And I think there's some enormous gains to be made there. I think we have to be very, very bold to take some big initiatives now by looking at things like improving photosynthesis. A really big, bold initiative. If you increase photosynthesis, you're going to increase productivity in the favourable years, in irrigated conditions, and you're also going to improve water use efficiency. Transpiration efficiency is photosynthesis divided by transpiration. So bold initiatives like photosynthesis is going to have an impact in all areas. I think we've got to really mobilise ourselves to take these bold initiatives. But I also think the root systems are important as well. And I'll close by listing a wonderful group of collaborators, people who I work very closely with, fantastic team, and I'll leave it there. Thanks. Pat.
Yeah, thank you for such a thoughtful talk. I think you were the perfect speaker to close this symposium.
Thank you.
I'd like to go back to one of your early points, that you think physiology can contribute to breeding by bringing in new variation. So I'd like to ask you, where have you gotten your variation? Has it been land races, synthetics, mutations?
Yes, all of those, Pat. I started my career in wheat looking at the diameter of the xylem vessel in the seminal root system. Sounds wow, what a way out, sort of crazy thing to do. But John Passura, a colleague of mine, is a very, very clever biophysicist, I guess, and he was able to, had compelling reasons to explain why changing the xylem vessel diameter in the young seedling root could be important, and it was important. 10% yield improvement. We couldn't find it anywhere in conventional gene pool, in the contemporary gene pool that breeders were using in Australia or whatever. We finished up having to go to a Turkish landrace to find that. Where in terms of our work for carbon isotope discrimination, we found that in a very old Australian wheat. In variation in Tillering, a landrace wheat from West Africa. So some of the dwarfing genes, about 4 or 5 dwarfing genes which we've been using, they're basically older, from basically older wheats or or results out of mutation studies. So it's been quite a mosaic of sources that they've actually come from. But the search has been exciting, and having the right phenotypic. So they've actually come from a number of different sources. There's no single source that I'd like to recommend to really milk. Yes, up the back there.
I'm very much impressed by your thought and very interesting and exciting. And I have one question to you. When your presentation, I understand that you are in favor of convention and breeding, and you say smart convention and breeding. But in other areas, like people also try to integrate plant breeding. So what do you mean by smart conventional breeding?
I think of smart conventional breeding as using the best ideas, the best technologies. It might be in statistics. The contribution of statistics in terms of spatial designs, in terms of plant breeding, in the last 20 years has been phenomenal. So adopting the spatial design, improving statistical power, and being able to remove spatial effects in the field. We saw the incredible spatial effects of Erie, and they're the same all over the world. And you can being able to remove some of those. Whether it's using double haploids, whether it's using single seed descent, whether it's using out of season shuttle breeding, there are multiple ideas that come to mind. Most breeders are pretty smart in what they adopt. They're pretty smart in where they put their priorities, and they're constantly evolving. Changes in statistics, changes in mechanisation. Mechanisation changes have been extraordinary. They've resulted in us being able to sow tens of thousands of plots rather than 1,000 plots, say. So it's taking the advantage of whether it's speeding up generations, shuttle breeding, whether it's exposing exposing the phenotype that you want by using specialised environments. Specialised environments for disease control, specialised environments maybe for acid soils, are smart ways of really getting at the phenotype and therefore the genotype that you want. So it's a combination of all sorts of things there, but it's quite exciting, really. You know, we tend to think of plant breeding as pretty mundane stuff, but my word, it's not. It's changing on a monthly, daily, almost daily basis. There are a lot of exciting things happening there. Yeah.
I want to get back to one of your slides, which you list the different component traits that contribute to drought tolerance.
Don't say drought tolerance. Water productivity. Sorry.
And you say that the best environment to select for those traits is this is my wife optimal environment. And that gets me concerned a little bit. One of the cliches is really is you always get what you select from. Should I be selected out for those component traits in the optimal environment? Should I be selecting for you the target, drought, town environment?
Yeah.
So that gets me kind of concerned at that.
Yeah, no, I think that you've raised a good point there. Taking a trait-based approach to breeding or a physiological approach for the traits that I think are important in my environment for my crop, it turns out that all of the ones that I list there, I can select for them more effectively in a favourable environment. But as a plant breeder in Australia, breeding in our dry environments, I would not be selecting doing all of my yield trials in an irrigated environment. I would be going into the target environment. So I think you've raised a very important dichotomy to what I've said, that for a trait-based approach, for a physiological approach, in my situation, favourable environments. But I think what you're saying is that if you're going to improve, if you're using yield as the integrator of multiple traits, then you should be growing it in your target environment, which is probably a dry one. I would avoid the disastrous ones, but I would be looking at an environment where I can select for yield potential, but also where I can select for some sort of adaptation to drought as well, as long as I can maximise the heritability. So that's a good point.
One quick question. The question about what productivity, which is that it's very, very relevant, and there is two parts of that one, the water, and that's what we're going to leave about the productivity of water. We discussed a lot about the crop side and the water side, which is the monitoring of the real measurement of the water in the ground, provided that we expose all our rudimentary into the soil profile full of water, and then ignore it. So that part, I think that is missing on your presentation. How do you go on that one? Because without having that water data, we will not have that benchmark value for how much of the dry water we will be getting. And we will never have the benchmark data on that one. I think your talk was very good in terms of the plant aspect, but the monitoring of the water, how are we going to go with that one?
How do we monitor water availability in the soil?
Yes.
Several ways. We use neutron tubes or TDR for that sort of thing, but the quick and simple and dirty one is biomass. The more transpiration, the more water use, the more biomass that you produce. And so the simplest method to look at how much water the plants are using is to basically measure what's the total biomass. Even though there will be some confounding with transpiration efficiency or water use efficiency, if you've got a line, well, what we're doing at the moment is we're growing plots where we actually measure biomass, where we can actually see whether we're using more water, we're using thermal photography to get instantaneous shots by getting an idea of how much transpiration is going on at the end of the season, also to get an idea of biomass as well. So there are a number of different ways, but you still have to probably use something like neutron moisture tubes to actually determine exactly how much water that you're actually using. Rashid.
I just want to go back to the question because I think it's one of the most important messages in the talk. This idea of selection environments for traits for yield. If we go back to Pasura's equation, those three components. Now, wondering about each one of them and where's the best way to, or the best environment. From my little experience I have on this, because for Delta, when you look at variability in well-watered environment, it doesn't necessarily reflect what goes on the dry scenarios. It's jumping. If you look at water extraction or transpiration, my feeling is, I don't know about wheat in your environment, but what they done in other environments, others, it's also pretty much variable. Water extraction in dry soil does not necessarily reflect the constitutive part of your system. Now, harvest indexes in our soil, and I think it's optimized. So what's your comment on that? I mean, I take the message from your experience, actually, it's very valuable in wheat, in your target environment. But can we really make this a general saying, okay, let's just look at traits in optimal conditions, or should we look more at the adaptive traits to drought? Because you have some crossovers or something.
I think I must be taking I'm interpreting this equation in maybe a simpler way or a different way to what you are. So there are three components. Improving any one of them is going to improve yield, or decreasing any one of them will decrease yield. So let's take a trait like leaf elongation rate. in dry conditions. Is that an important trait for dry areas? Okay, leaf elongation rate. Will it increase harvest index? No. Leaf elongation rate. Will it increase water use efficiency? No. Leaf elongation rate. Will it increase water use? Well, maybe, yeah. Maybe it will. Maybe it increases total leaf area. So yes, okay, that's something that could be important. Not going to increase harvest index, not going to increase. So it's like a framework to think about the traits, to think about the genes. So even though you say, in your experience, carbon isotope discrimination might flip-flop around the place, although that doesn't happen in our experience, if we measure it at a particular stage, but you can say, okay, water use transpiration efficiency or carbon isotope discrimination. Harvest index? No. Water use efficiency? Obviously. Water use? Maybe, because it changes stomatal characteristics and so on. So I think the way in which it's very useful to use is by looking at your target trait, by looking at your favourite trait, by looking at your favourite gene, you can say, now, how will that gene or how will that trait change one of those three components? And if you think, well, it's not going to change that or that or that, give it away, I say.
Well, the question was more about the environment. In which environment should they look at it?
Well, your target environment. Yeah, your target. So for me, it's easy. My target environment is Australia, and this makes it very easy for me because I can think, well, this is going to work in the western part of the country, but it's going to be a total failure in the eastern part of the country. So I'll just target it on genotypes that succeed in the west. So you have to think about in your target environment.
I got a good message from you. You are saying carbon 13 is a good parameter to select the yield. And I saw most reference we use for the delta carbon with carbon 13 subdivided by carbon 12. So based on your experience, can you tell more about why you choose carbon 13 instead of carbon isotope discrimination?
I'm not sure if I understand your question correctly, but you're asking about the value of using carbon isotope discrimination.
Yeah, you have slightly mentioned select carbon 13 content.
Yeah.
Not delta carbon 13.
Well, carbon 13 content is actually a component of the of carbon isotope discrimination or delta.
Yes. Carbon 13 itself.
Oh, itself.
Will show more variation than carbon isotope discrimination.
I disagree. Maybe we can follow that up later on. Roberto.
I'll go back to what we were discussing with Rashid, and I think a very good example that I also tend to agree that I would probably prefer to put my eggs in the basket of the environment that maximizes heritability, that most of the time is bound to be the environment with higher productivity or better conditions. If you look at Maine, all the improvement that has been made with mainly the work of Don Dubik shows that very clearly. You showed that in one of your slides. Most of the improvement in maize under low water available conditions has been come along because of the selection of breeders done under optimal conditions. I think perhaps the tricky part, going back to your flip-flop or whatever, I think it depends what sort of direction you want to go in the trait you want to select for. So maybe I'm giving you a little bit of an extreme example, and I think carbon isotope discrimination is one perfect example, because also Australia, it has, if you grow your wheat and store the moisture, you have to go one direction in terms of what you have to select for. But if you have grown your wheat in another part of Australia where there is rain, then you have to go in another direction. So I think the key point, theoretically, particularly for traits so highly heritable as carbon isotope discrimination, you could select in one environment going in the opposite direction, knowing that you have to pass in the other environment. But I think the key point, rightly so, And it's also true for any QTL study. And I like, my mantra is phenotyping is king and heritability is queen. So if that happens to be under highly productive environment, well, you may have that crossover interaction, but that doesn't tell you that even in the favorable environment, if you know you're going to have the crossover interaction, you can decide to select in the opposite direction. Yeah, I think
Know your trait, know your environment.
yeah.
Very important.
And the other thing, I think it should be clearly said here, and this is I use a lot of the Pasteur equation, but we should not forget that these three components are not independent one from the other. And that may have some very dire consequences if you don't keep that in mind.
Sure.
Because you may increase one, but you may kill the plant to death because the other components are not going to come back.
Sure. It's just a framework to think.
Yes.
Question from Renee.
One of the things that I know I found difficult in thinking about water productivity, perhaps working with woody crops like maize and rice that have a really high fruit, a fruit of very unusually high sensitivity. And so when you take, I can take the exact same amount of water and get quite a decent yield or nothing at all based on when the stress occurs. Have you seen any ideas about how we could come to a number that's meaningful, that incorporates that concept of the stress-sensitive period?
Yeah, that's an interesting point, Renee. And it's one which I haven't thought a great deal about because wheat is probably a more robust species. I suppose but I can imagine that there are catastrophic events in any crop which are going to destroy that relationship. It might be a catastrophic event of drought and reproductive time. For me, for Scott, it might be frost or something of that sort, which is a catastrophic event in a wheat crop. So I guess it remains true in the absence of a catastrophic event. When there is a catastrophic event, then, well, you could, when there is a catastrophic event, it probably doesn't hold as much.
Or maybe you calculated during that period or something.
Yeah,
like that. Maybe
yeah.
you come up with a convention that would be
Yeah.
It will create an envelope curve.
Yeah, that's right. Yeah.
All right, I have another question because it goes back to what you were saying that I absolutely agree. We have to look more below ground. But first of all, I want to ask you what you mean below ground, only the root or also the rhizosphere? Because my impression is the rhizosphere is so complex, it can't be handled from a scientific standpoint because it is not reproducible experimentally, it means we cannot handle. The second point that I would like your enlightened opinion, we haven't discussed much about osmotic adjustment. Particularly if you look in wheat, which is a strong osmotic adjustment, at least as compared to maize. What about, and also the contribution of possible land races or wild species that may bring in this component, adaptive component, for which hopefully the plant will not have to pay a price under what water conditions. Because osmotic adjustment may have a price. So, have you, in your experience as a breeder or talking, after there's been a lot of work on osmotic adjustment, what is your feeling? Is it a dead horse
My understanding of the situation with wheat is that, again, it might help you get through a catastrophic event. Yep.
In terms of root growth, not necessarily.
Yeah, I get back to Renee's point of the cost of growing roots in terms of osmotic adjustment. I think, but I think I think there's clearly a cost there, and I think the point that Renee made is a very, very valid one. The amount of exudates coming out of roots is just phenomenal. It's just fantastic, I think, really. Being able to bind up aluminium, being able to solubilize phosphorus, being able to kill off different pathogens in the soil or whether it's, there's just such fantastic stuff going on below there we know nothing about. I don't think I'll say anything more other than it's a very, very fertile ground, and I'm sure everyone in this room probably agrees about what a fertile ground, the below ground root system really is. Yep. Maybe it's time to close it off, John, is it?
All right, well, let's thank Dr.
Oh.